Right, people, please chat with me. Can you hear me clearly? Come on. Come on, send a message. Can you hear me clearly? Yes or no? Come on, come on. Send a message, somebody. Come on, please. Can you hear me? No? Yes, perfectly audible. All right. Okay, thank you very much, Noha. Thank you very much. I think you're the only one, right, who knows to message, right? Network drops. Right, people will go on discussing the tutorials. We have two tuts, MSc tuts remain to discuss. Right, I have two tuts to discuss. We'll start with people MSc 29. MSc 29, just give me 1 minute. Right, people, let's take those two tuts. Can you find people tutorial 28, but it's actually 29. MSc 29. This one. Can you find it? This one. Right, people, 29. First question, how many years ago that prokaryotes have evolved on Earth? How many years ago that prokaryotes have evolved on Earth? What are you going to write, people? 3.6. 3.6 billion years ago, right? 3.6 BYA, you write it full, yeah? Billion years ago. 3.6 billion years ago. Next one, mention structural features of them. What are you going to say, people? Now you know that they are, please chat with me, students. You know that they are prokaryotic, anaerobic, heterotrophic, but they are also structural. They are also structural. So you can write only 1 point then. What is it? Prokaryotic. Others are not structural. Prokaryotic. Prokaryotic. Right? Prokaryotic. Next one, mention the abundant elements in addition to carbon, hydrogen, oxygen, and nitrogen in living matter. What are the other elements, people? We can take phosphorus, potassium, calcium, sulfur. What we said, Chopkins? Chopkins Camag. Chopkins Camag, right? Chopkins Camag. So this is, you write the remaining part. So we'll take it like that. We'll take it like that. ඔයා මේ සෙටිං එකක් ගහනවා මේක ගහන්න එපා මෙහෙම. ගහපුවම කරදරයක් නේ resolution උත් නෑ අන්තිමේදී stream වෙන්නෙත් නෑ. නෑ පා දැන් හරි. දැන් හරි ඒක දැන් වාත වෙලා ගිහින්. දැන් ඔය end. නෑ බජා මේ ඒ අනවශ්‍ය සෙටිං එකක් නෑ නෑ. මෝඩ සෙටිං ගහන්න එපා. හරිද? මට සාමාන්‍ය දිනේ සෙටිං එක දාන්න බැරි නිකන් ඉන්න. යන්න. Right, people, next one is mention the relevant carbohydrate. Mention the relevant carbohydrate. Polysaccharide found in middle lamella. What is it, people? Pectin, eh? Pectin. Linear structural polysaccharide with glucose as monomer. Linear structural. So what is it, people? Cellulose. Next one, storage disaccharide with ketose. What is the storage disaccharide with ketose, people? What is the storage disaccharide with ketose? I can take, people, storage disaccharide with ketose. What is it? Anyone knows the answer? Storage disaccharide with ketose. Can someone tell the answer? Right? Storage, they say disaccharide, so it can't be maltose or lactose. So this has to be what, people? This has to be sucrose. Good. Sucrose. One student is chatting with me. Sucrose. Fructose killadi, but that's wrong. Sucrose. Fructose is not a disaccharide. Right, next one, people. Mention the cause to consider phospholipid as an amphipathic molecule. What is the reason, people, for the phospholipids to consider amphipathic? What do you call amphipathic? Polar, non-polar, both features present in the same molecule. We call that as amphipathic, right, people? So you can write that the hydrocarbon chain has hydrophobic. We'll write it like this. Hydrocarbon chain has hydrophobic and phosphate group, and the attachment is hydrophilic. Phosphate group and attachment is hydrophilic. Right, next one, mention the nitrogenous bases which are paired with two hydrogen bonds. Nitrogenous bases paired with two hydrogen bonds. You write in words, adenine and thymine. Adenine and thymine. Right, next question, people. Mention two differences during observation of the specimen in light microscope and electron microscope. Two differences during the observation of specimen in light microscope and electron microscope. Right, first one, in light microscope, specimen is directly observed. Electron microscope, it is not directly observed. Right? We'll write that. Image directly detected in light microscope, but it is not detected directly, not detected directly in electron microscope. In electron microscope. Not detected directly in electron microscope. Number two, light microscope, living and non-living, both specimens can be observed. Observe, but in EM, specimen non-living, specimen non-living. Specimen is going to be, it's non-living. Right. Then third one, people, they ask two differences. You can write some more. Actual colors visible in LM, actual in LM, black and white in EM. Black and white images are visible in EM. Then you can see that the light microscope, we use dyes to stain heavy the specimen. Heavy metals are used to stain specimen. In light microscopy, dyes are used to stain. In electron microscopy, we use heavy metals. Stains are used. Heavy metal stains are used. Next one, what are the causes for the genetic variation? Now they ask only two, so I think you have enough space to write. I wrote 4 in that same space. Hari? Question number 5, people. What are causes for the genetic variation in meiosis? You can write 3 reasons here. One is lamai crossing over. Crossing over. What is the other one? Recombination. Recombination. Crossing over, recombination. Yeah, independent assortment. That's good. Independent assortment. Independent assortment. My next one, people. Next one. Next one, part B1. Define centrosomes. Define centrosomes. What are centrosomes, people? They are the region near the nucleus where a pair of centrioles present. Right? We can take that as the centrosome. Region near the nucleus. Region near nucleus where a pair of centrioles arranged perpendicular to each other. Perpendicular to each other. Right, people. Next one is mention functions of cilia in human body. What you can say functions of cilia in human body? Cilia. Where do you find cilia? Some reproductive cells have cilia. Move fluid over the tissue surface. Yes, in the respiratory passage they move. Right. Now some people are chatting with me. Couple of other people are chatting with me too. Very good. Very good. Hari. Oviduct help to move egg. Now this Zoom chat system is now a little different, people. They have changed the settings so I can't see your chat continuously. Instead, I like click and see that. You this new changes I don't like. Mention functions of cilia in human body. Move fluid over surface. Move fluid over surface. Right. And what is the second thing? Help to move egg towards the uterus. In oviduct, help to move egg towards uterus. Towards uterus, right? Next one, people, mention the main components. Mention main components of extracellular matrix. What are main parts of the extracellular matrix, my dear? Glycoproteins and other carbohydrate containing molecules. I think you have to say that glycoprotein are the carbohydrate containing molecules secreted by the cell. Right. Moving to next page, my dear friends. Right. Right. How ECM, extracellular matrix, affects for the cell behavior. How ECM affects for the cell behavior by involving mechanical and chemical signaling. Mechanical and chemical signaling. Right. Next one, define cofactor. What are cofactors, people? Non-proteinous components. Non-proteinous components. Non-proteinous components which are essential for catalytic activity. Essential for catalytic activity. Catalytic activity of certain enzymes. Of certain enzymes. Of certain enzymes. Right. Right. Next one, people. Is my the image clear or not, people? This image is clear or not? The video is not clear. For me, it's not very clear. Is it clear for you? I want to go to the board, but this poor internet connection today, it drops all the time. I think it's this setting, so I'll fix this right for next class. I think I need some update to my studio now. Okay, we'll see. Right, shall we move to next question, people? In which circumstances that ATP act as allosteric inhibitor? In which circumstances that ATP act as allosteric inhibitor? You have to say that when ATP exceeds the demand. When ATP exceed the demand. When ATP exceed the demand. What is considered as P680 in photosystem? What is considered as P680 in photosystem? 680 is a reaction center which absorbs 680 nanometer wavelength light. We'll say that reaction center. Pigments, neither. Pigments that absorb 680 nanometer light. And next one, how excited photosystem 2 is neutralized? How excited photosystem 2 is neutralized? What happened, people? The excited photosystem is neutralized by the taking the electron release from hydrolysis of water. Right there, electron release as a result of hydrolysis of water. Electron release from hydrolysis of water. Next one, we will mention two steps of Calvin cycle. Two steps of Calvin cycle that need ATP in addition to formation of 1,3-bisphosphoglycerate. Two steps of Calvin cycle that need ATP in addition to formation of 1,3-bisphosphoglycerate. Regeneration of BioEve. Regeneration of RUBP. Regeneration of RUBP. Right, part C1. Mention the criteria based on genetic materials in present classification system. Mention the criteria based on genetic materials in present classification system. Mention the criteria based on genetic materials in present classification system. Genetic materials neither on present classification system. So what are you going to write there? First one is the sequence of bases of DNA of important genes. DNA of important genes. What are the other one, people? The sequence of bases of DNA in chloroplast and mitochondria. Sequence of bases of DNA of mitochondria and chloroplast. Mitochondria and chloroplast. Right, next one. Mention the usage of keys. Mention the usage of keys. Now, you know, people, we use a dichotomous key. We use the dichotomous key for the grouping of organisms and for the identification of them. We'll write that to group and identification. To group and identification of organisms. And next one, mention two domains which same response to antibiotics. Now, what are the two same domains that responds to antibiotics? Mention two domains with the same response to antibiotics. What are you going to write, people? That is archaea. Right? That is archaea and eukarya, right? Archaea and eukarya. So, what they ask people? Mention the two domains. You write the domains, archaea and eukarya. Then they ask people and mention that response. When growth not inhibited. Growth not inhibited to streptomycin and chloramphenicol. Streptomycin and chloramphenicol. Next one, mention the polypeptide, mention the polyphyletic group of organisms. Mention the polyphyletic group of organisms. We can take people Protista. Protista is the polyphyletic group. Why they are considered as polyphyletic? Because origin from more than one ancestor. Origin from more than one ancestor. Next one, mention three main features of dominant plant of Lycophyta. Three main features of dominant plant of Lycophyta. What are you going to write, people? Three main features of dominant plants of Lycophyta. What is the dominant plant? Sporophyte. So they want you to write sporophyte features. Terrestrial and some are epiphytes. And some are epiphytes. Terrestrial and some are epiphytes. What else you can mention? Sporophyte, dominant plant. Sporophyte, dominant plant. And then upright stem and ground hugging stem. And ground hugging stems present. Ground hugging stem present. Right. Then people mention two ways of getting nutrients by gametophyte of Lycophyta. Two ways of getting nutrients by the gametophyte of Lycophyta. What do you want to say, people? One is by forming symbiotic fungi, right? And photosynthesis. Photosynthesis by symbiotic fungi. From symbiotic fungi or fungal association or by symbiosis. Next one, people, mention the phylum with the following features. Mention the phylum with following features. Branch gastrovascular cavity. You can take two phyla. What are those two phyla? Mention the phylum with following features. Branch gastrovascular. Now, gastrovascular cavity present in two. Branch one is present in Platyhelminthes. Platyhelminthes. Second one, endoskeleton with plates, Echondamata. Echinodermata. Then body covered with tough cuticle that undergoes ecdysis. What is that? Nematoda. That is nematoda. Right? Okay, that's the tute MSC 29. It's actually given as MSC 28. Can you correct the number? Can you correct the number as MSC 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 29? Okay, MSC 29. Right, and then people, there is MSC 30, MSC 30 also, and there is a CAM paper too, ne? CAM 34, CAM paper 34. I think we can take CAM paper for the next class, ne? We have time, so we'll take CAM paper for the next class. Shall we quickly go with the other MSc paper? The other MSc paper, kiyada lamai 30 ne? MSc 30. MSc 30. Next week, people, can we have a paper 6 was unable to join class last week. Hurry. Paper 6. Yeah, submit paper 6. Hurry. Right. Let's take these questions, people. What are disaccharides? What are disaccharides? And let's check that part. What are disaccharides, people? Sugars formed by joining two monosaccharide by glycosidic bond. Can you write there? Sugars formed by joining two monosaccharides by glycosidic bond. Sugars formed by joining two monosaccharides by glycosidic bond. Nine. Then give two functions of disaccharides. Storage sugar of milk. Storage sugar of milk. Sugar of milk. I want to know sugar cane. Sugar cane. What else you can say, people? Translocate sugar in the phloem. Translocate sugar in phloem. Translocate sugar in phloem. Name the chemical groups of phospholipids that makes the hydrophilic head. Chemical group hydrophilic head, phosphate group and its attachment. Phosphate group and its attachment. And next one, how saturated fat differ from unsaturated fat made up of saturated fatty acids are saturated. What you can say? Saturated fat made of saturated fatty acids. Then again, unsaturated fat made of unsaturated fatty acids. That's enough, ne? Fatty acids. Right. Next one, people. Which are physical agents that make the denaturation of protein? Now they ask physical, don't write chemical and all. So you can write temperature, high temperature, ne? High temperature, and high energy radiation. High energy radiation. Now don't write anything else, detergents, acid and all, don't write them. Indicate the mitotic phase of each of the following events. Kinata ko attach මොකක්ද ළමයි ලියන්නේ? Prometapase. Prometapase, right? Spindle microtubules depolymerizes. What is that, people? Telophase. What is the malignant tumor? Tumor which is invasive and attack one or more organs. Tumor which is invasive. Tumor which is invasive. What do you call invasive? Attack, neither. Invasive and attack other. Attack one or more organs. Right. Moving to next one, people. B. How do you indicate the spread of the cancer? What do you call with the one name? Metastasis. Metastasis. Give the basic basis for the present system of classification that you know, right? Sequence of bases of DNA of important genes. I'm not going to write that. Then sequence of the bases of DNA of mitochondria and chloroplast. Base sequence of ribosomal RNA. Base sequence of ribosomal RNA. Any problem? No. Base sequence of ribosomal RNA. Sequence of amino acids of common protein. Sequence of amino acid of common protein. Then other molecular structure details of cellular components and all you can take. Other molecular structural details you can take. Then which group possess book lungs? Arachnids. Next one, good. Which of the animal phylum consists only marine echinodermata? Right, students, moving to the other side. Right, complete the following dichotomous key based on the following species given: pogonatum, vascular tissue present, vascular tissue absent. Vascular tissue absent, pogonatum absent. Nephilopis, cucurbit, Caecus, coconut, Marcantia absent. Vascular tissues present. Stem leaf absent. We'll take that vascular tissues present number 3. Vascular tissues absent number 2. Leaf stem stem leaf present nang pogonatum ne. Pogonatum. Right. Stem leaf absent nang marcantia. Marcantia. Plus a homospores. Then number 3 ne homospores neprolepis, cucurbit, caecus, coconut ne homospores present out of the remaining ones. Homospores absent. Kaudlamai neprolepis ne. Homospores absent. Number 4. Then cone present. Cone present. Who has cone? Caecus ne. And cone absent number 5. Then pollen grain contain 3 pores. Pollen grain contain 3 pores. And pollen grain do not contain 3 pores. How do you take that? Pollen grain with 3 pores. Kauda and 1 pore. Coconut neither. Right? So we'll take it like that. Pollen grain contain 3 pores and 1 pore. Dicot neither. I cannot remember now that. Can you 3 pores dicot ne? 1 pore monocot. Pollen grain with 1 opening monocot. Yenang coconut. Then 3 pores, what do you say, people? Cucurbit. Cucurbit can mukada. And cucurbit can, what is that, people? Cucurbit is pumpkin, ne? Cucurbit is pumpkin. Cucurbit pumpkin. All right. Then moving to next part, people. I think we have to enlarge it this little more to see it clearly. Right, what is P? Okay. What is P? P can what am I? Primary xylem. Eh? Q. Secondary xylem. R. Vascular cambium. S. What is S? Secondary phloem. T. What is that? Primary phloem. You can't see that in your picture, right? So we can see once it is enlarged only, you can see it clearly. Even I fail to recognize these parts, right? Looking at that tiny diagram. Too small, eh? Too small. Right, then people identify the above diagram transverse section, TS, transverse section of secondary grown dicot stem. Secondary grown dicot stem. Name PQRS, we did that. Done. Briefly describe the structure found in U for the gaseous exchange. What is that U? Lenticel leather. So you have to describe the lenticel. Small pores present in periderm. Now, we said, what are that people? Horizontal slits kada kyun? They are horizontal slits present in periderm, small pores. Small horizontal horizontal pores present in periderm. Packed with loosely arranged coke cells. Packed with loosely arranged coke cells. Next one, what is water potential What is the people? The physical property that predicts the direction in which physical property that predicts the direction that predicts the direction in which water flow governed by solute concentration and applied pressure. Applied pressure. Right. Water, what is water potential value of pure water? What is water potential value of pure water? 0 kilodala megapascal kilodan. Otherwise wrong, yeah? 0 megapascal. Which light receptor is responsible for this part you haven't done yet, right? We have to do it. Blue light photoreceptors, we have to do it, right? So then that's all for the day. Thank you very much for your participation. Right? You have paper 6 to answer. Right? Do paper 6 and then submit that paper 6. Sir, paper 6, please. Paper 6, paper 6, paper 6. Wait. Right. Collect paper 6, submit it to the website. You know how to do it. Matakala Karandone, right? You know the threatening last time. You have to create the folder, upload it, and I'll see you then soon, right? In weekend class. Thank you. Salute for your participation today. Bye bye. Good night.